Ready for a professor hunting adventure?
Yes, very much so.
Good man, hello. So Jaya, we've left Leamington Spa.
I know. I'm very excited about today. So, hi everyone. It's been a long time since my last video log, and it's great to be back here talking to you again. And we're particularly excited because we're going on a trip to London to meet the scientific director of Anthony Nolan, no less. So we are very excited, and to add extra spice to the whole thing,
Has he got a name?
yes, his name is Professor Madrigal.
The professor. It's the doctor. Okay, yes, sorry, carry on.
So he's yeah. So we'll talk about him a bit later. To add extra spice to the whole proceedings, the beast from the east, also known as Siberian weather, has hit the UK. So you may not be able to see, but we're driving in sort of driving snow, if you like, hurtling towards London, and I'm very pleased that my good friend Giles has got the madness to get me through this weather. He'll get me there, whether it's using just a shovel and a piggyback, but that adds extra spice as to whether we'll get there or not.
Absolutely.
But no, we're really looking forward to this trip.
Yeah, good one. And the end of cabin fever, Jaya. You've broken out after months.
I have escaped. Yes.
Thank God for that.
Yes. So
how many months have you been sort of trapped around treatment and home? Basically, necessity.
On and off really since September.
Yeah.
So I mean, yes, I've got out, as it were, but this, for the first time, I've got no specific treatment planned. They're monitoring me to see how the last few interventions are going to work. In essence,
but you're still mad. It's not really worked.
No, not from that point of view. No, it's not helped brain function. But I'm still here.
Yay!
And so no, we'll talk about what happened to me of late.
A little bit later.
Yeah.
And the other thing is, you've got a rather special new walking stick to show off to us today, Joe.
Well, yes, I can. It's one of the side effects of the treatment is that it's damaged my nerves, so I've needed a walking stick. So that's I look even more crazy than usual, actually, with the walking stick.
They're quite distinguished, I think.
Do you think so?
No, I do. I think I was a bit disappointed there wasn't a silver head on the cane, though.
Well, yes, we were looking for a sort of silver skull, but we just came up with a standard NHS wooden curve top.
Okay, well we'll do that. We're keeping down with the kids, down by London.
Yes.
All right, nice one. All right, Jaya, here we go. Off to London.
Yeah, great.
Cool, man.
Hi guys, this is very exciting. We finally arrived in time, I would have to say, with just 1 minute to spare, typical Giles, at the Anthony Nolan Institute. So
Yeah,
very excited.
Excellent. Okay, here we go.
Hi, hello.
Thank you so much for holding the door.
Thanks. Okay.
Go on in, Jaya.
Hello.
Hello.
Hi.
Nice to meet you.
Nice to meet you. I'm Jaya. Hello.
Very nice to meet you.
Lovely to meet you too.
Oh, nice to meet you.
Into the bosom of the heart.
Thank you. Hello, Professor Madrigal. I'm Jaya Ravindran.
Nice to meet you.
Very nice to meet you.
Very nice to meet you too.
So I had been reading about you, and obviously I'm very glad that you are under Nala Ravin and you're responding very well.
Yes.
Excellent. Very good.
Let me say firstly, it's an enormous privilege and an honour to meet you. You are the most decorated scientist, doctor, human being I think I've ever
How
met.
do you know that?
Well,
You did some search.
I've done a bit of research all the way from America to London, and having been here for the last 25 years.
5 years.
I just wanted to say, firstly, a very big thank you to Anthony Nolan and you for all the work that you guys have done. Without you guys, having been a bone marrow recipient and recently had a donor lymphocyte infusion, I wouldn't be here. So I owe a big, big thank you to all of you, as well as my donor, of course. But without the Anthony Nolan Institute, I really wouldn't be around like a lot of other of my fellow patients. I think one of the things while I was doing a bit of background reading was how ignorant I was of the spectrum and diversity of research that you do. I wondered whether, as a kick-off, you could give us an overview of the type of research that Anthony Nolan does.
Okay. So first to say that the reason why we exist here as an institution is because patients like you need us. And we have a very enormous privileges to be able to try to help patients like you. And following the mission of Shirley Nolan that tried to save Anthony, establishing the first registry in the world. And by doing that, we have been helping other registries worldwide to expand their registry, the number of donors. And now, thanks to all this effort, we have over 31 million donors worldwide, which is amazing when you think this is altruistic people willing to save the lives of many patients. And that has helped us to provide a large number of donors, and with innovative developments in other areas like cord blood transplantation, and now haploidentical, what we call haploidentical plus, we could say that we could find a donor potentially for every patient in the world. But saying that, still 50% of the patients, unfortunately, don't succeed to the outcome of the stem cell transplantation, mainly because there are three main problems. The disease come back, relapse. The second reason is potential infection and what we call infection. So all the graft versus host disease is another complication. So there are several things that we need to work very hard to do that. And it's amazing how the war sorts of effort the medical and scientific community has focused in trying to resolve these problems. And now more than ever, we are very close to do that. There are new innovative treatments, CAR T cell therapy, regenerative medicine, advanced cell therapy, where we modify genes and modify cells with gene modifications, and we can do that. So Anthony Nolan has been focusing from the beginning in trying to save lives. And when I was appointed scientific director, or I came from Stanford, as you say, 25 years ago. In fact, this week, I think we will celebrate a little bit of that. We obviously start this place, this institute, and a small place that somehow has contributed worldwide to innovative science in many areas, and to train a large number of people in the field. We had many students have graduated here and have been receiving degrees, and that's because we also want to help the global community and train people. So we have people from all over the world who has come to this place. It might seem a small place, and yet it has made an impact in many areas. It has contributed to develop innovative technologies for typing optimally the donor and the patient, for developing ways to expand T cells and other cells from cord blood or from other areas that now we could try to do cell therapy. We had worked with the medical community, with the patients in the field. So I think we still have a lot of things to do. And I think we need to work very hard, and that's what we try to do every day, because still the challenge is quite big, and it's obviously trying to be sure that when patients like you receive a transplant or any other way, we could actually offer the opportunity for them to have a higher rate of relapse. Ideally, we shouldn't need transplant patients. We should be able to use, as many disorders are now being treated with drugs or with chemo or with some medications. The idea is that we have to tailor optimally the medicine. So it's going very well, but it's still a very big challenge.
Yeah, No, thank you. One of the things, when I worked as a rheumatologist, we had several markers or biomarkers of, let's say, progression of a particular type of disease. How close are we to having that sort of technology in hematological malignancy? So, let's say, predicting outcomes, predicting things like graft versus host disease, graft versus leukemia effect. How far are we from that sort of, and how far are we from predicting outcomes prior to transplantation? You know, using the sort of studies that you've done.
I think, as I mentioned before, we are now in a stage, in a new age, where we are really using new technologies, what we call next generation sequencing or third generation sequencing. We are learning a lot about what we call polymorphisms in some particular genes that are more oncogenic. We have also start to understand the mechanism of some combinations, some recombination between genes. And thanks to that, we can start to use new drugs. In fact, the condition that unfortunately you have, TAL, has been associated to some mutations, for instance, in Notch1. And this is a kind of recombination that comes from two chromosomes, 7 and 8, that produce a kind of rearrange of a protein that is associated to sorts of transport of some vesicles. The fact that we identify patients that has these mutations plus other associated mutations to that make them sort of, we can say that there is a better prognostic if you have those mutations positive. And also there are new drugs that people are trying to identify how we can modulate, by particular Notch1. Notch1 has also been associated with Alzheimer's. So there is a lot of investment trying to produce a drug that can actually modulate that. But another example, which is the most sort of a stronger and pragmatic illustration of how innovations in understanding mutations can help to treat, is what happened with chronic myeloid leukemia. Chronic myeloid leukemia was the main condition for stem cell transplantation in unrelated bone marrow donors until about 15 years ago, when the development of a new drug, imatinib, has helped us to block the mechanism of recombination between BCR-ABL, what we call a Philadelphia chromosome. And now these patients receiving treatment, oral treatment, can be, if not cured, they can be treated with no need of transplantation. So the development of new markers are very important, but that's in terms of the disease. In terms of the other question that you asked, in terms of predicting graft versus host disease, or in terms of predicting other conditions, I just came back yesterday from a meeting in the United States, which is the American Society of Transplantation, and I'm going next month to the European bone marrow transplantation meeting that will take place in Lisbon. I was the president of this society for 5 years, and what has been reported in this meeting and in the abstracts that will be presented in the next month is that there are a large number of new proteins or variations of genes that are helping us to see the mechanism of graft versus host disease. And because that, there are now new drugs, monoclonal antibodies, that block some particular products. For instance, graft versus host disease is very strongly associated to the activity of some cells that we call Th17, which the mechanism of action is through a particular protein, which is it's called interleukin-6. Blocking interleukin-6 block a little bit the process of inflammation, and that's just to mention something. But it's still predicting which patients are going to develop GVHD or not still is an obscure area. And we are working, in fact, here, one of my students is trying to understand the mechanism associated to the GVHD at the level of genetic variations. And we are doing this in collaboration with several hospitals, and we hope that that could help us. The other thing that has been obviously an enormous advance has been the matching of donor patients. We here at Anthony Nolan have developed technology, currently we call third generation typing sequence, that help us to understand the degree of matching completely. And by doing that, we hope to provide, as much as we could be able to do, an optimal donor for every patient. An optimal donor might not always be the perfect match, might be the right combination of genes or mismatch that might help us to come with the outcome. But we are now in the process of analyzing that data. So I think there are several things that are going to help us to, if not make every transplant successful, increase substantially the benefit of many patients' outcome.
It's certainly, I remember, well, the few papers I could understand, the title, let alone, the actual science behind it, thinking it's not just simple as HLA matching, there are epigenetic factors, there's peptide binding, there is the interaction of the T cells as the conductor of this sort of immune orchestra, if you like, and how that interacts with the whole complex. And it just, one, I found it fascinating, two, I didn't really understand it, but three, it just shows the challenges that we have in actually trying to get the best outcome for patients. One of the things that I was particularly interested when you were talking is graft versus host disease, and how it's a double-edged sword between graft versus host disease and graft versus leukemia, and how to get that balance right. Is the research in any way able to try and differentiate between how to optimize one and reduce the other, if you understand what I'm saying?
No, of course. We assume that in a healthy individual, the immune cells, in particular some cells that we call T cells, T lymphocytes, are practically patrolling, going around our body and recognizing any abnormal cell. And by a normal cell, it could be an infected cell or a cell that in the process of division might have acquired mutations that could lead to tumor expansion. We believe that our immune system in some ways keeps us alive, removing tumor and malignant cells, recognizing some particular abnormalities that we call tumor-associated antigens or viral-associated antigens. In fact, you receive DLI, donor lymphocyte infusion, and we believe that the actual transplant is the mechanism by which we eliminate the tumor cells. Obviously, the patient will receive chemotherapy or radiotherapy, but there are always residual cells that they will grow back. It's actually the graft, the cells in the graft that are going to be coming from a donor, related or non-related donor, that is capable to go and recognize those residual tumor cells and eliminate it. And that's what we call graft versus leukemia. The problem is that we don't know exactly what are the markers in most of the cases that are responsible, the targets, to eliminate those cells. What we know is that, for instance, when we do autologous transplants, when you get the cells from the same patient and you remove, let's say you purge the tumor cells and you provide only these T cells, because already that immune system didn't work in the first instance and let the tumor escape, if we could call it like that, we see a large number of relapse. If these cells, you are providing an army that already was incapable to recognize the criminal, let's say, or the enemy. And by doing autologous transplants, we see a large rate of relapse. When we do a perfect match sibling donor, we also see a certain degree of higher mismatch than we would expect when we provide an unrelated donor, because there are some more difference between the donor, unrelated donor, and the patient, which has, as you say, the risk of leading us to graft versus host disease, but also has the possibilities that those differences might be the mechanism by which the cells from the donor might eliminate the residual tumor cells in what is called graft versus leukemia. The border between graft versus host disease and leukemia is still one of and graft versus leukemia is still very uncertain. And we see, for instance, that patients that for some reason might develop graft versus host disease in the process of the transplant, which in some ways a little bit of graft versus host disease is not bad. When we see patients that have degree 1 or 2, we see less chances of relapse. And that's in general. We don't want too much graft versus host disease, but a little bit is convenient. When the patient goes after transplant with not graft versus host disease, it's very likely that the patient could relapse. And so it's a little bit of cross-border. But when we, on one hand, try to modulate graft versus host disease by giving them immunosuppression, the chances of relapse are again higher. So we reduce the immune system and the patient relapse. So what we have as a challenge is identify tumor-specific antigens that are not associated to sorts of matching donor cells and leads to graft versus host disease. The new innovative therapies with CAR T cells, a new mechanism by which you get the cells from the donor, or hopefully, and people are working to use an unrelated donor, you get the cells, these T cells, and now you put a sort of cassette of modified genes inside, and that cell now expresses a protein on the surface of the cell that is a kind of antibody that recognizes a particular protein. In the case, for instance, of ALL, it recognizes a protein called CD19, B cell, which is a protein that is present in all the B cells, the lymphocytes. But because the tumor cells express massively these ones and are the ones that divide very quickly, it will stick to them and kill the cells by a mechanism associated to the machinery of cell recognition between the T cells and the target cell. It might also wipe the normal B cells, but it removes the tumors. And we are seeing amazing results with that kind of treatment. And I think there are still things to improve, but it will be a very good mechanism to treat many patients with this kind of condition. Identifying targets that help us to direct those cells or modify cells to other kind of tumors or leukemias, for instance AML, has been more difficult. And we are working in the community all together to do that.
Yeah, no, I think it's I find the whole graft versus host disease and graft versus leukemia a bit of an enigma, because when I had my relapse after bone marrow transplant, it occurred my skin relapse occurred at the same time as an episode of graft versus host disease, suggesting that the graft versus leukemia effect is less effective for maybe extramedullary disease and relapses than medullary relapses. I don't know what you feel about that.
It is something that still is unclear why some people even relapse in certain areas that are not systemic, and so only we see the disease in a particular area that come back, and why it's not a systemic one. And probably, and I don't know exactly the case, but probably when you started to have graft versus host disease, probably you were immunomodulated by using cortisone or trying to control the GVHD, and that could actually led to the relapse. It's very difficult to know because sometimes both conditions might don't be targeting the same antigen. It might be completely two in parallel process that you see graft versus host disease and in addition escape of tumor, because it might be that graft versus host disease might be recognizing a particular target antigen that doesn't have to do anything with the tumor cells.
Okay. Now, one of the few papers I actually could understand was, I think you did a thematic analysis of donors, like a sort of qualitative paper, looking at what motivations donors had, be it altruism, determination, etc. Now, what can we learn from that research which would help me as a fundraiser and someone who wants to raise awareness in my community? Because I know that despite all the campaigning and whatever that's done, although there are only 15% of males, young men, on the register, they are responsible for 54% of donations. And also if you are a patient, you know, you obviously have a 1 in 4 chance of having a match with your siblings, but and a 60% chance of having a match with a stranger. But that falls to 20%, I think, when you're from an ethnic background such as myself, you know, black, Asian, ethnic minorities. That was one of my initial worries, that I wouldn't find a match, and I was so lucky to have had so many matches. What do you think are things that I can do or we can do to try and improve the uptake of the registry?
I think it's very important what you are asking. You highlight in first instance this paper that we did, but behind that is one of the most beautiful things that we found in our everyday life work, which is the enormous sort of the optimal human kindness in terms of being able to donate a bone marrow to risk is this beautiful altruism and this sort of need to try to help our human beings, friends, so in people who even we don't know. And that's what 31 million people worldwide are trying to do by becoming members of the panel. What we see, as you say, is the majority of these numbers are mainly Caucasians. So when you search for a donor, as you say, will be very likely that when we search for a Caucasian, we will find 80% chance to find a matched donor. It depends on what kind of Caucasian, but in general, or which country, but in general it's success. But as you say, minorities is different. And an effort has been done in UK and globally to try to resolve this problem by trying to raise awareness and understanding of the significance of becoming a donor. So education, as that paper reflects, is one of the knowledge and being aware that they can actually, everybody can save lives by being sort of capable to provide a bone marrow and stem cell, which is in general a very safe procedure, or even a cord blood is something that is a wasted material after the baby is born. What we found is that certain communities have, for lack of knowledge, religion, and cultural issues, not so much understanding of what is the implications and the capacity that they have to help other human beings. So I think it's very important to work with the community, to work in every aspect, in the early ages of their life, in the education, in the school, and also to show the significance of how other communities are helping to save patients like you. I think you were, as I understand, your donor came from India.
That's right.
And that was one of the challenges that we actually, Professor John Goldman, the former and late Professor John Goldman, who was actually the medical director of Anthony Nolan, really understood that the only way we could actually try to help a large number, for instance, of patients like you, was to work in each country to try to help to develop local panels. And in fact, I had the opportunity to go to India and work with the local authorities and groups there to start the first registry. That was some years ago, and I love you. India is a fantastic country and culture and everything, but it was amazing to see how people can be, once that they are informed and understand what happened, the first registry in India was developed, and that's amazing, and thanks to that, patient like you can be helped. So we will never be able just to raise the number of donors in UK that are necessary to save patients with your background, or black Afro-Caribbeans or Spanish people. We need to work in the communities too. But also here is to work with them, to show what we need to do, to educate them, to make them participate in everyday life. And that's what the paper reflected in that information. And I think that's a major task that Anthony Nolan has, and as you know, we had been working a lot with the communities in different ethnicities, trying to increase their awareness.
And I know one of the papers, I think if you did, was to work out about how much cord blood you need to form a bank. Now, translating that to the registry, do you have an idea of a target of how many people we need, critical mass, if you like, of in the registry, so that we can optimize our matching?
again, it comes to what kind of community or ethnicity we are working on it. When we look at the cord blood has potentially some advantage, which because it's a more sort of an immature immune system, or it contains some cells that are not so reactive, we see less graft versus host disease when we provide some degree of difference, what we call mismatch between the certain proteins that we call HLA, which are the critical ones to match between donor and patient. It's like a barcode that we always try to match between donor and patient. And there are some lines in this barcode that we call 10 of 10 or 8 of 10 when you match only 8 band of 10. And in cord blood, we can do transplants with a higher degree of mismatch, 1 or 2. It's always optimal. It's better to do 10 of 10.
They're more immature cells.
That's correct. And still, we don't know exactly how to make every cord blood transplantation very efficient, but it seems that statistically we see less graft versus host disease, chronic graft versus host, acute and chronic, and we see less relapse. It's just that because it's a small number of cells, it takes longer to engraft, but it's a good source of stem cell. So we did a paper some years ago and estimated that comparing the population of donors that we have and the population in UK, and we came that with 50,000 cord blood units, we could be able to, with some degree of mismatch, provide to find cord blood for 80% of the patients, so it's no higher in UK. That number has been sort of readjusted, and we believe that with 30,000 at least we can afford substantial numbers of this. The panel is potentially, we would say 10, our donor panel, and our panel is about 700,000, and in UK with the combined is over 1 million donors between the different registries. And still obviously have to struggle to find that one. As you say, for certain communities, Asians and other ones, is only between 10 and 30% chance to find a potential donor for some of them. And that's where we had to work to expand those ones. So you put everything together, we could talk about millions. It depends, for instance, if you are looking, some communities are more, some ethnicities are more diverse.
Yes.
But for instance, Afro-Caribbean communities are more diverse, and therefore the number of donors that are required for matching or to find a perfect donor for those patients might be substantially larger.
One of the studies you did was on Mexicans, and saying how there was within Mexicans there were so many subgroups of ethnic origin.
I'm glad that you have read so many papers. In fact,
I'm trying to learn the headlines because actually that's all I could all the ones that I could actually understand.
Anthony Nolan has published a large number of papers. I don't know, I would say including abstracts and everything, we can talk about 1,200 papers. So now you bring papers that we wrote, and I think they had been changing, some of them the practice in UK and globally. For instance, we published data that shows that patients that receive a donor under 30 years old has 10% better chances of survival or outcome, better outcome. So therefore that helped us to change the registry. And we work also on the demonstration that saliva was a good source of DNA, and we started with the spitting, and now we use vocal swabs. That is a normal way to get our typing of donors. So certain papers might be very dramatic, and we will publish them in Nature Genetics, as we did when we invented a technology called RSCA, Reference Transconformation Analysis. Some other papers will be simple, but change practice, as you say, in terms of understanding the diversity. That particular paper that you mentioned, I am Mexican, so a large number of my students had come from there, but also we had been working with the communities in different areas that help us to understand this diversity. These proteins that I mentioned to you that are important in matching are very are very polymorphic. There are thousands already identified, but we believe that there are still, it does represent a very small number of the total enormous polymorphisms, enormous difference that exist in the population. We have a set, all our cells have the same kind of barcode, but when you look at the population, the numbers and recombination of these 10, 12 genes that you have can be as large as millions and larger than the total human population. So we need to find ways to match and to find that particular donor, or what people are trying to do is to go beyond the degree of matching. That's why people are using cord blood, where we can actually allow some degree of mismatch, or what we call haploidentical plus, where we can take a related donor, but that is not a perfect match, but there might be one haplotype identical, and that's why we call haplotypes. Only half of the genes of the father or the mother will help us to do that. But then we need to manipulate the graft post-transplant by giving some drugs that will eliminate some of the reactive T cells. So I think the community is working globally first to try to increase the number of donors. Second, to try to increase the representation of donors from different ethnicities. And if that's not possible, also to come with cord bloods or banks of cord bloods that represent and has a high number of them from minorities. For instance, we in Anthony Nolan has focused our collection in areas where we can have more larger number of peers of the community, where the bears would come from patients from different ethnicities, and that has helped us to increase and provide donors for those ones. And third is to go beyond the matching when we cannot provide that. So we have the haploidentical, we have other ways to do that, and potentially, which is coming more as a source of new development, is to come with innovative cell therapy that could help us to target directly the tumor.
Engineering T cells and natural killer cells.
Perfectly. Yeah, I mean, you have a condition which is on T cells, but similar condition which is on B cells, the ALL, already is the target of CAR T cells.
Yeah.
So
now, one thing you mentioned is potentially we need millions on the register. What would your views on perhaps having a compulsory, opt-out scheme for transplant? Rather like they're proposing for solid organs in Wales, for example, I think.
I think obviously we all have the opportunity potentially to if not safe, to provide a graft or opportunity to make someone's life much better. I mean, patients that die, unfortunately, they still could provide organs, eyes, or different organs to save their lives. And that's something that in some ways require again education. In Spain, for instance, is almost compulsory, and the number of transplants using post-cadaver organ is quite impressive. Kidneys, heart, liver. I think we need to bring that globally. But that's a different kind of graft. The unrelated bone marrow donation is mainly when you are alive, and you need to be educated. It has to be altruistic. It cannot be actually mandatory or anything like that. It should be because you need to be convinced that that's what you are going to do as a donor. One of the main problems that we face at the registry level is having potential donors that we enroll, and then when we go to try to ask them to donate, either we don't find them, or they are actually no longer willing to donate. And sometimes it's understandable because they might be in a particular phase of their life that might not be the right time to be donating. But on the other hand, we want to be sure that people understand the commitment that they are acquiring and the enormous responsibility, but at the same time, the privileges that they are acquiring to save life. It's interesting, and that paper reflects that when you ask them post-donating, they will do it again.
Yes.
And they are very pleased to have given the opportunity to save the life of somebody. And if you ask them to give extra cells, they will be happy to do that. So that's the kind of donors that we need.
They demonstrated distress, didn't they, when the patient didn't do so well. But I was just thinking, well, if it is a matter of education, then by giving them the opportunity to opt out, that would provide a window for patients to debate in their minds, would I be happy to commit or not? And then it might be an opportunity for for more people perhaps who are more altruistically minded than not. Do you see what I mean?
Yeah, I agree, and it's that, but also cultural, as I say. I mean, even for instance, donating a cord blood require education to the community. Some people are immediately concerned that might be a major problem, when it's not. It's a tissue that, as I mentioned, will be discouraged, but yet we find sometimes consent for donating not very much substantially approved. And that related also to different ethnicities and cultural. So it's not just religion, it's also cultural. So that's what we need to work on the community to do that.
Yeah. I mean, I think the message that I'm trying to get through to everyone, and I'm trying to use religious places as hubs for communities, is it only takes a mouth swab, a saliva swab, to register. And in 9 out of 10 cases, it is similar to giving blood. Because I think people still have that notion that it's a medieval process where you drill into bone to extract it, when in fact, in most cases, it is just like giving blood.
And some people even believe that bone marrow transplantation is giving bones to somebody. It's not just even giving stem cells. So as I say, it's a lot of education. But it's very important that people understand the long-term commitment, that when they actually provide saliva, they hopefully will be forever in the registry until they can, until we have the chances to need. Obviously, there is certain age that we don't use donors, but that's not so important. It's the commitment.
Well, that's been a real. Thank you so much for talking to me. I feel very humbled and privileged to talk to you, Professor Madrigal. It's been. I don't know, I'm just so grateful, and for all the work that you guys have done, and the reason why I'm here is because of Anthony Nolan, so I feel very motivated to give something back, not just to Anthony Nolan, but as a doctor now, as a patient to my fellow patients, but I feel very grateful. Thank you so much.
Thank you, and just to say that contrary to what you say, I'm very privileged to be shaking your hand.
Yes,
I had been all my life dedicated to try to, as a doctor and in this position as director of as scientific director of Anthony Nolan, to try to be sure that every patient like you have the most optimal transplant or the better of the chances of survival. So for me, it's an honor to have you here, because in some ways, it shows that what we are trying to do, our battle, although still we need to do a lot of things, somehow is helping us to win little by little.
And that's the message I want to give. It's a real privilege for me to feed back and give hope, which is so important.
I think one of the most beautiful things that the human being has is the chances of giving. It's the only thing that we have. Give love, give a smile, give part of yourself. And if you can give something that saves a life, it's really a privilege.
It is special, isn't it? It's that golden thread of humanity that binds us together. It's just
it is. I mean, even a smile makes such a difference for so many people, and we sometimes don't understand that giving is the most beautiful gift that we have.
May I ask a question, Professor? What would be the question that you would imagine your peers would most like to hear you answer?
In the field?
Yes.
I think it's a tricky question. Obviously, it depends what you cover. If it is in terms of how, as was put, how we can actually control graft versus host disease, how we can make tolerance, because stem cell transplantation can help to save many other conditions, not just malignant disorders or non-hematology. We could probably try to offer this procedure to treat patients with rheumatology conditions, autoimmune conditions, and once that we start to understand how we can provide the optimal donor without having the complications, what we call transplant-related mortality, then actually, if it was so low that has much benefits that giving a stem cell transplantation to a patient, for instance, with lupus, then we could actually do that. So how we can do that? That's an answer that all the community has to work. But also, the other area is how we can work with the community globally to make every transplant, every donor, first people to be aware and conscious of what we do. Because no, you don't have to be a stem cell donor to save the life. You can be a supporter, you can be someone who works with the community, educating, training, and other areas. So it's a tricky question, because I could answer that question to you in different ways. I don't know if I
Well,
you wanted to hear something else.
No, it's always interesting to see what someone thinks. I mean, I imagine when you're a leader in your field, you're one of those rare people where you type into Google an institution and the name that comes up is you. I mean, that's not often that happens, and so we are honoured to be here. And thank you for your time. And you're a lovely person to listen to.
Thank you.
Cream rises to the top, and I can see how you got there. But I just wondered, sometimes within one's peer group, they would love to see in your mind and go, I wonder what he's actually thinking about that. Or, you know what I mean? I wondered if there was a twinkle in your eye almost where you would say, well, there is just one thing I was thinking about. Is there anything that comes to mind when I say that?
No, I think it's what you say. I think it's bringing the best of every human being. I think we all have something to do, and I think that's the spirit that we need, the Renaissance.
Absolutely. I think Jaya had something he wanted to give
in that
you.
spirit of giving, I wanted I've written a poetry book.
Oh, I read about that, and I'm very keen because I love poetry,
Okay, well, I was going to give you a book. Now, I don't know, Giles, whether you've got a pen.
I have not a pen.
Sorry about this.
No, I will be honored. I wrote a novel myself.
Have you?
And many of these paintings are mine. That ones, that
We
ones.
have been filming them. I was wondering whose they were.
So those ones that were mine.
Sorry, which one's over here?
Like these ones, all these ones that you see there, that sculpture is a little bit mine. And I wrote a novel, but I don't have it with me here. But the cover is this one. It's called Days of Rabies. It's unfortunately in Spanish only, but it will come.
Lovely. You wrote a novel. So is it something people are buying?
It is in Spanish, but I am trying to get it translated.
Right.
It's called Días de Rabia, Days of Rabies.
Days of Rabies.
Or rabies,
Do you write when you're on holiday, or do you write?
I write sometimes at the airports, in the planes, so I write in hotels. So when I have some time, I sleep very little, so I have the fortune of being able to work very early in the morning, some extra things.
Yeah. Well, you work hard. You fill time. Other people just sit there lazing about or reading a paper, probably.
Here you go,
This is
sir.
an honor,
Okay.
and I'm very glad to receive this. And no, it's an honor and a privilege just for me. Thank you very much, honestly.
All right. Thank you very much. Really appreciate
Thank
it.
you. No.